Life begins quietly. There are no dramatic sparks or thunderclaps, just a single microscopic cell that flawlessly divides into two identical copies. That simple division repeats until the result is an adult human body, comprised of roughly 37 trillion individual cells. But before a single cell can split, it faces a monumental task. Hidden deep inside the nucleus, it has to completely and perfectly duplicate its entire genetic code. The human genetic blueprint is built from billions of specific chemical letters, all packed tightly into a microscopic space. If the copying process drops a single line of that code or inserts the wrong letter, the resulting daughter cell won't know how to operate. It will be missing the basic operating instructions required to survive. The ability to multiply safely requires absolute precision. Flawless genetic duplication is the silent, ongoing prerequisite for the existence of all complex life. To see how a cell pulls off this massive duplication, we have to look directly at the target, the DNA double helix, a long molecule locked together like a spiraling ladder. When James Watson and Francis Crick modeled this unique structure in the early 1950s, the physical shape immediately hinting at how it might duplicate itself. Their model revealed a dense, interlocking puzzle of chemical rungs, but it also posed a mechanical problem. The genetic information is locked inside that twist. Before anything can be copied, the cell must physically pry that locked ladder open to expose the inner sequence. To do this, it deploys a specialized molecular engine called helicase. The helicase acts like a wedge, driving straight through the center of the molecule. It unzips the bonded strands and pushes them apart, creating a Y-shaped opening known as the replication fork. By tearing the structure in half, the cell turns a closed vault of information into an open, active template ready to be read. With the two original strands separated, the cell is left with two half-ladders. The challenge now is to construct the missing sides. Enter a new piece of molecular machinery, DNA polymerase. This builder enzyme latches onto the exposed strand, pulling in free-floating genetic building blocks from the surrounding environment. It reads the original chemical sequence letter by letter. Whenever it sees an A, it plugs in a T. Whenever it sees a C, it plugs in a G, assembling the brand new continuous side. Because the two sides of a DNA strand physically run in opposite directions, this building process is asymmetrical. One side is synthesized smoothly in a single continuous glide, while the opposite side has to be assembled backward in choppy, disconnected fragments. When the polymerase finishes its run, the process yields two complete, identical DNA double helices, where there was previously only one. Notice how each of these new structures is built. They each contain exactly one strand from the original parent molecule, bonded to one brand new synthesized strand. Biologists call this a semi-conservative process. Nature's method of preserving half of the old code to directly guide the assembly of the new code ensures absolute fidelity. The template itself acts as the ultimate safeguard against copying errors. But this biological machinery operates at lightning speed, adding hundreds of letters per second. With billions of connections to make, mistakes are a mathematical certainty. Every so often, the builder enzyme slips and forces the wrong genetic letter into the sequence. These tiny copying errors are what we refer to as mutations, a direct change in the core code of the organism. To combat this, the cell maintains an active defense force. Specialized proofreading enzymes follow right behind the builder. They scan the fresh strand, cut out the mismatched letters, and plug in the correct replacements almost instantly. Without this constant editing, the stakes are dire. If a spelling mistake permanently slips past the proofreaders, it can cause genetic instability, leading the cell to malfunction or triggering severe disease. Yet, the system works. This machinery unwinds, copies, and proofreads billions of letters, performing flawlessly trillions of times across the span of a human life. This semi-conservative replication is the biological bedrock that preserves our traits, ensuring that life can safely and accurately pass from one generation to the next.